Hello everyone. Last time we see the classification of the enzymes. Now moving towards we will see each class one by one. So apart from the six classes, the first one is the oxidoreductase, which is denoted with the EC number 1, that is the enzyme commission number 1. Now we'll see what is meant by the oxidoreductase. So on the basis of the name, oxidation as well as the reduction take place. So here it deals with that the enzymes bring about biological oxidation as well as the reduction between the two substrates. So the biological oxidation and the reduction between the two substrates, that we call the oxidoreductase. For example, A and B, these are the two substrates. So in between these two, the oxidation as well as reduction take place. Next, this class again subdivided into the following subclasses as I shown here. One by one we will see which are the simple one. First is a dehydrogenases, on the basis of the name that it removes the hydrogen. Okay, so see, so it catalyzes by the removal of the hydrogen from one substrate and pass it to the another. So here the two substrate, so one substrate remove the hydrogen and it will pass to the next one. Example, malate dehydrogenase. Next, oxidases. So in this the enzyme use the oxygen, so that's why the name is given oxidases. Example, tyrosinase. Third, hydroxyperoxidases. On the basis of name, enzyme which use hydrogen peroxide as a substrate, that we call hydroxyperoxidase. Example, peroxidase itself. Next, oxygenase. Here enzymes acts on the single hydrogen donor with incorporation of the oxygen directly into the substrate. So see here the two functions. First, it will act on the single hydrogen donor. How? By the incorporation of the oxygen directly into the substrate. Example, tryptophan. And the last one, hydroxylases. Hydroxylases, here the enzymes act on the pair hydrogen donors with the incorporation of the oxygen into one donor. So compare these two. Oxygenase, here enzymes act on the single hydrogen donors. Here enzymes act on the pair hydrogen donors. Here there is incorporation of the oxygen directly into the substrate. Here incorporation of the oxygen into the one donor. Example, phenylalanine hydroxylases. So these are the five subclasses of the first class, that is the oxidoreductase. Now moving toward the second one, now we'll see that is the transferase. So see, transferase, that is a transferring enzyme which is denoted with the enzyme commission number 2. Now we will see regarding the transferase. So first, what it means, the enzymes that catalyze the transfer of some group from one molecule to the another molecule. So here see the example. So this X group is transferred from one molecule to the another molecule. First it is towards the A molecule, now it is towards the B. So transfer of the some group from one molecule to the another, that we call transferase. So it again divided into the following subclasses on the basis of transfer of group. So first one, transaminase. On the basis of name, it includes transfer of amino group. Aspartate aminotransferase. Example is transphosphorylase, transfer of phosphate group, that is the hexokinase enzyme. So in the presence of this hexokinase enzyme, the phosphate group is transferred. Transglycosidase, transfer of glycosides. Example, phosphorylase. Transacylases, transfer of acetyl group. Transmethylases, transfer of methyl group. Now it's a simple one. The third class, that is the hydroxylases, which is denoted with the enzyme commission number 3. So what it deals exactly, see, hydroxylases, the enzymes which bring about the hydrolysis are called hydrolyses. Okay, so enzymes on the basis of its hydrolysis, that we call the hydrolyses. So it again subdivided into the following subclasses. Simple one, esterases. Example, lyases. Peptidases, example, pepsin. Amidases, example urease, glycosidase, example is the amylase. Okay, so these are the first three classes of the enzymes. Now moving towards the next one, that is liases. So the liases, what it means? So liases, it is denoted with the enzyme commission number 4. So these are the enzymes that catalyze the mechanism other than the hydrolysis. So the liases, it is that enzymes which catalyze other than the process of hydrolysis and leaving the double bond. Okay, so here the formation of double bond in this enzyme. So it again divided into the following some classes on the basis of their linkage. Okay, so on the basis of their linkage, it get divided into the following sub classes. Carboxylases, example pyruvate decarboxylase, aldehyde lyase, aldolase, carbon oxygen lyase, fumarase, ammonia lyase, histidase. So these are the four subclasses of this lyases. In these all, the double bond will be formed. Isomerases with the EC number 5. The enzymes that catalyze interconversion of some isomers. It may be the optical, geometrical, are called the isomers. Means there is an interconversion